So now that the stage is literally set for the speakers, we are going to start this first session, and we have very strong team, and then strong team means strong openers. So our first speaker is Dr. Thangaraj, who is also the organizer, and everybody knows he has made phenomenal contribution to human genome variation studies, particularly to our population structure in this country. And he is going to talk about human diversity and its relevance to health disparities. So before Thangaraj begins, so I will ring this buzzer at 20 minutes, so which means speakers know that you have now 3 minutes to wind up or early, so that we have 5 minutes for the question answers. And so allotted time is 30 minutes, so 2 minutes will be for this kind of change of stage and other things, right? Okay, Thangaraj.
Thank you, Rakesh. Again, I welcome all of you, and good evening. So when you talk about human diversity, there is no other better place than India, because even phenotypically we see a lot of diversity. There are people who typically look like Africans. Some of them typically look like Europeans. Some look like Southeast Asians, and some are very unique to India. Therefore, there is a huge amount of diversity exists in Indian subcontinent. What is the reason for such a huge diversity? Our earlier studies suggest that one of the population, which is in Andaman and Nicobar Islands, which is about 2,000 kilometers away from the mainland, particularly from Chennai and from Calcutta. There are 4 tribal groups, as you see here. That's how they look like. By looking at the complete mitochondrial DNA sequence, we found that they have very unique sequence. These are the evolutionarily conserved mutations, which is not seen in anywhere in the world. Therefore, they have been assigned a new haplogroup called M31 and M32, and based on that, we have concluded that probably they are the first modern human who migrated out of Africa about 60 to 65,000 years back. Since then, they are in that particular island. Of course, some moved further to Australia. The Australian Aboriginals, Andamanis, and some group in South India still now share genetic similarities. After this, we used the nuclear markers, about 1 million markers from Affymetrix. Based on this study, we concluded that we know that these two are very deep-rooted, African and Andamanese divergence. And while they were migrating to Andaman, some group stayed back in southern part of India. We call them as ancestral South Indians. And we predicted that probably there was another wave of migration from Africa, and there was a divergence from somewhere in the middle. One group has gone to Europe, other group has come to northern part of India, whom we call as ancestral North Indians. There are several subsequent studies where we have shown that Europeans and ancestral North Indians have genetic affinities. To give you a few examples, one of the Y chromosome haplogroups, R1A, which is predominant in Europe, also seen predominant the same frequency, almost same frequency in northern part of India, but not in southern part of India. So yet another example, the mutation which turned the skin color from black to white. We studied the gene called SLC24A5. Is again, you can see it's predominantly present in Europe, Middle East and northern part of India, but not in southern part of India. Another example is the lactate persistent gene, which again the mutation helped in digesting the milk sugar when we become adult. It's also predominant in Europe, Middle East and north or northwestern part of India, but not in South India. I told you about initially there was two major founding groups. In addition to that, there are several other migrations. For example, invasion of Alexander army, military conquest of Arab and Turks, British colonization. Then some of the recent migrants like Siddhis, Jews, Parsis, and so on. To understand each one of them, to look for their genetic root, we have shown the Siddhis who have been brought by Portuguese traders during 15th to 19th century, who are found now in Gujarat, Karnataka and also in Hyderabad, but of course the Hyderabad ones are totally admixed, therefore we didn't include them in the study. And we trace back and they are part of the Bantu speaking Africans. Of course, mostly the males have come, and if you look at the mitochondrial and Y chromosome haplogroup sharing, you can see the Y chromosome mostly from Africa and mitochondria from Indians. But if you try to trace the other population, Jewish who are in Mumbai, Cochin and also in Calcutta, but we analyzed Mumbai and Cochin Jews, and we found the traces of them. These are the one, as you see they're all Indians, and you can see the Middle Eastern component is present in this Jewish population. Of course, we found that the Jewish who are in Cochin are of 5th century and Mumbai are much older, 7th century. Then the Parsis, that was recently we have studied, Parsis who are living in different part of India and Pakistan, and we again found that, so these are the Parsis. You can see they have the component of the one which is in the Middle East, suggesting that there were several migration into India, and we are clearly seeing the evidence of their ancestry. Very recently, by looking at the bone samples of Harappan region, the only bone which was been sequenced, and found that they have a genetic component of both the early human who migrated to India, the Andamanis, and the Iranian hunter-gatherers. And the age was about 2000 to 3000 BCE, but of course it's not a very old bone sample, but this again suggests that this has the very old genetic component, and there was a migrant, of course, was very old, also suggesting that the agriculture probably have started independently in India. Yet another study, by looking at more than 500 bone samples of the Middle East and looking at the samples of contemporary populations from Central Asia, South Asia, and we have shown that the Amazon migration, in fact you can see that they have migrated towards north, then come to India. Probably they are the one who brought the Indo-European language, and they were also migrated to Europe. Now if you recollect that initially what we presumed, that because the people from north and Europe, they share some kind of genetic affinities, it's very clear that the people have migrated from Africa in the second wave, and after some time people migrated to India and also to Europe. So all this put together makes India as one of the very complex country, where we have more than 4,635 endogamous groups, including large number of caste population, large number of tribal groups, and some of them still practice hunter and gatherers lifestyle. As you see here, 36 of them are still practicing hunters gatherer lifestyle. That means they don't do any job including agriculture. So what is disparity in this? Particularly health disparities. Let me first look at India as a whole, and no other better study from us than this. Of course, Dhandavan is here, he may talk more about this. We have shown one of the genes which are associated with cardiomyopathies, myosin binding protein C3 gene. Of course, there are a large number of mutation has been identified. One of the mutation in the intron, 25 base pair deletion, it skips away the wild type exon, therefore there is a truncation of the protein. Hence, it leads to sudden cardiac death. If the mutation is in homozygous condition, it leads to sudden cardiac death at very early age, less than 10 years. As you see here, the postmortem tissue of the children who died less than 10 years, dilated and hypertrophic cardiomyopathy. But if it is in the heterozygous condition, the individual live up to 40 or 45 years without any symptom, but after that, sometime they collapse while doing severe exercise or running or doing cycling and so on. When we try to look for this mutation in Indian subcontinent, so far the normal individuals which we have from different part of the country, looking at more than 6,000 samples, we found on average in India there are about 4.5% of this mutation in heterozygous condition. When we try to look for this mutation in different part of the world, to our surprise we found that the mutation is restricted only to South Asia but not in other countries. And later on we found this mutation arose in India about 33,000 years back, and our human diversity study helped us in understanding that, because initially we shown that there were two groups, probably they have admixed, and during the admixture it got well spread throughout India and nearby countries. Because whatever we say about India is also true for South Asian countries, because India contributed about 50% of the genome to our nearby South Asian countries. Similarly, another gene, RAF1, which again causes the childhood cardiomyopathy, is again present. Of course, it's very low frequency in India but not in other countries. So these are some of the mutation. It's well spread because it causes disease, the first mutation, only after 45 years when it is in the heterozygous condition, therefore it's not well spread. But there are some diseases which are restricted to certain community, are restricted to certain geographical region. For example, these are some of the population. One is Handikudu. Mustaks also here who collected the sample. And you see that it is restricted to that region. There's a place called Handikudu in Shimoga district. Most of the individuals in the village are all of very short stature. There's a bone abnormality. Similarly, there's a high frequency of nephropathy in and around Vizag in Andhra Pradesh. Most of them undergo dialysis. In addition to that, through our clinical collaborators, we get to know that there's a population called Raju community in Andhra Pradesh. There's a predominant frequency of cardiomyopathy. Particular arthritis in Reddy populations of population of Andhra Pradesh. Alkaptonuria in gypsies from Tamil Nadu. And Vaishya has a very unique problem of pseudocolon esterase deficiency. Because of that, the regular anesthesia cannot be given to them. If it is given and they may not metabolize the anesthesia, they remain in the anesthetic effect for a very long time, and it may lead to paralysis. Similarly, Madras motor neuron disease restricted to Chennai and nearby place, sickle cell anemia in Chhattisgarh and some other region. There are many such population-specific, regional-specific diseases are existing in Indian subcontinent. So what is the reason for such population-specific diseases? Our earlier study have shown that there are two founding populations, one is ancestral South Indian and ancestral North Indians. And to understand that when did that admixture between these two groups took place, we analyzed large number of samples from South and North, and we come up with this kind of hypothesis where these two founding populations have gave rise to many groups. They have evolved independently. That was happening for the last 4,000 years, and for the last 2,000 to 4,000 years, these two founding groups have admixed, the exchange of genes between these two, and you can also recollect the cardiomyopathy mutation, myosin binding protein C3 gene. After admixing, for the last 2,000 years, each population started maintaining the endogamy. That means marrying within the group. Therefore, whatever has happened genetically in each group remain within the group. Maybe the caste system which arose around 1,500 years must have put more pressure on maintaining such endogamous marriage. To understand that, what is the impact of the endogamous marriage? Again, we analyze large number of samples, 2,800 samples from 260 populations from India and nearby South Asian countries. And we found that most of the Indian population or South Asian populations are having a high IBD score, identity by descent. You can see there are two populations which are highlighted in red color. Each line represents one population, Ashkenazi Jewish and Finnish population. And these two populations have been well studied. Large number of disease has been mapped, and most of them are recessive disease. But look at Indians. Large number of Indians are with several fold higher IBD score. Now you can imagine what kind of problem most of these populations are having. I believe that most of the information about their health conditions are not even come to the attention of medical community. There are a few examples, and before that, to tell you in simple way that the IBD score is that if the populations are maintaining endogamy, and you expect that if there is a recessive mutation, The mutation causes only when it is existing in homozygous condition are going to be high among each of these population groups. How that happened? If you look at this pedigree, for example, there's a mutation which is recessive in nature, therefore there is no disease here, and she married somebody who doesn't have the mutation, then she passing that onto the next generation, both the daughters receiving this mutation. Again, it's an heterozygous condition because both of them are marrying somebody outside, so it's passing on to the next generation. After a few generations, if they are living in different villages, they really don't know, or they must have lost the contact, they become unrelated. That's what generally happens. If you go into village, there are 100 families, all the 100 must have come from a single parent. But now they say that they are unrelated, but they are from a common founder. If there is a mutation, the founder is going to be there, most of the individual. But if these two individuals who are carrying this mutation in heterozygous condition, if they marry, that's when there's going to be a disease. So that condition is very, very high among Indians. To give you example, from our other study, the mitochondrial diseases, there are large number of diseases which are caused by either mutation in the nuclear gene or in the mitochondrial genes. Yeah. So in this case, what you see here, the mutation is not in the mitochondrial DNA, but the mutation in the nuclear gene called polymerase gamma or POLG. This is a wild type. In the parents, it exists in heterozygous condition, as you see here, CT and CT. Of course, this is a consanguineous family. They are absolutely fine, 45 years, 39 years. But you see, unfortunately, two children who received one mutant chromosome from the father, one from the mother, and this child died, and we don't know fate of this child. I'm sure this also is not alive now. And this mutation, which is in the homozygous condition, it introduces large number of mitochondrial DNA deletion, and these are some of the markers which use for diagnosis of mitochondrial diseases, and there's a lot of lesion on the brain. And the other family, this is unrelated family, but I'm sure that this mutation is from common ancestor. What you see here, 41 years, 38 years, absolutely fine, and both the children unfortunately have died. We don't know how many such cases are in the rural area, where if one or two child or children are dying in the family or few families, I'm sure that's not going to go into the medical community or to ICMR or somewhere else. Therefore, it's very, very clear that the endogamy leads to large number of recessive disease, which we are planning to study now. Finally, just one study which I wanted to show, that there's also a problem with the migrant population. Particularly, there's an example from our own study, where one of our collaborators from Institute of Neurology at University College of London, the symptoms are looking like it's a mitochondrial disease, but the mutation which generally they see among the Europeans are not existing. But they did exome sequencing and found the mutation in RNase H1 gene. Then he asked us to check whether this mutation is also exists in our cohort of sample, but unfortunately we didn't find. Then I asked, where from this? He said that the samples are from India, can you check it? But we didn't find, because most of our samples are from South India, and I asked him, where this family has come from. He said they are from Gujarat, all the three families. Yes, that's quite evident that what we have seen earlier, that each population is very unique. Unfortunately, we don't have any samples from Gujarat or Northwest India to find out whether this mutation is prevalent in that population or not. Therefore, it's a very clear evidence that the human diversity is always linked with health disparity. With this, I will stop. I am sure that our colleagues who are going to speak in the coming session going to highlight more of that. I would like to thank those who have contributed, particularly Dr. Laljit Singh, my mentor. We have lost him very recently. A lot of my students and of course Rakesh for the recent collaboration and setting up the Genome Center, and a lot of collaborators within India, and I am sure that some of them are here, Professor Satyamurthy, Mushtaq, and many more. The list goes, the Navani and many more. I'd like to thank the funding agencies, CSIR, the major funder, UK India Education Research Initiative, DBT, DST, and MRC, that's Medical Research Council, London. Thank you.